Okay, so this is going to be an extension of the things we talked about last time, which is non-allelic homologous recombination, which is to say that recombination, of course, involves sequences that are similar or identical to one another, and they are homologous because of that fact. Allelic means that the sequences occur at equivalent positions on homologous chromosomes. So these are, of course, very similar to one another, and recombination is a normal process that does not cause any problems usually. Okay, the problem arises when we have so many repeats in our genome, which are similar to one another in sequence, but are not allelic to each other. So such recombination between such sequences can occur and can have very bad effects on our genome, on our genes. So this is also called illegitimate recombination because the sequences are similar, but they're not actually allelic, even though the sequences are sufficiently similar to allow homologous recombination. So just to illustrate the fact that this can be dangerous to our genome, here's a table of some diseases that have resulted from this sort of recombination, mostly involving ALU repeats, but also some involving L1. So not to think that all these diseases are always caused by this type of recombination. For example, breast cancer causing mutations in BRCA1, there's many different mutations in that gene that can lead to a predisposition to breast cancer, and only this one that we're talking about has to do with ALU. But nonetheless, there are many examples where this type of recombination has led to defects in genes, ultimately to the creation of genetic disease. Okay, so last time we talked about equal sister chromatid exchange, and I will reiterate a little bit about that this time. More importantly, though, we're going to talk about intrachromatid, which is to say exchange involves repeated elements which are within the same chromatid. And there's two ways in which that needs to be considered. One is when the repeats are in the direct orientation, which we'll see, and that leads to deletion. And the other form is intrachromatid exchange, which involves sequences that are inverted, which is to say pointing in the opposite direction. And those lead to inversion of the sequences between the repeats, with bad effects sometimes. So especially non-allelic homologous recombination involving transposable elements can occur because there's so many of them. It's kind of amazing that we don't have more of a problem with this than we do. Okay, now, lots of times when you can see something like a LTR that's just sitting there in the middle of nowhere, you don't know how that arose, right? The example of intrachromatid versus sister chromatid exchange, both could lead to that same outcome. So you don't always know what happened, but you know that something did to lead to the existence of a solo LTR. So I'm going to give a couple of examples where that's happened. Okay, so remind you what a retrotransposon looks like. It has a certain genomic region that encodes its protein coding genes, but it's flanked by LTR, which is long terminal repeats, one on each end. And there are many examples in our genome where we do not have full-length retrotransposons or endogenous retroviruses, but rather instances where just the LTR is just sitting there in the middle of the genome. Okay, so one way in which that can occur is, as we've seen before, with unequal sister chromatid exchange. So what happens is the two LTRs will line up, but out of register. Instead of the first one aligning with the first one and the other end aligning with the other end, they line up such that the first one is aligned with the last one and undergoes recombination. So when that happens, you can have the formation of a fusion chromosome, which actually has two copies of the transposable element with an LTR in the middle, or a solo LTR. So to follow these recombination events, start on the left of the chromatid, go to the right. When the X occurs, go to the other chromatid, and then continue. And so you can see the outcome of these recombination events. And this is the same as we've seen before for sister chromatid exchange. The intrachromatid exchange mechanism is rather different. It will involve the two LTRs on the same chromatid. In the first example, where we have direct repeats, and that leads to deletion of the region between them. So it's not so easy to align these sequences because they have to occur in the right order, as you'll see. If it's ABC, we have to line them up as ABC. And that is going to involve twisting the molecule around to allow the homologous sequences to pair with each other. So we twist the molecules around, and then we do the recombination and look to see what the outcome is going to be. So here's one representation of this, where the little red things with the arrows are a directly repeated element, meaning the letters are ABC, ABC as before. Now to get those to pair up such that they're pointing in the same direction as each other, you've got to twist the chromatid around. But once you've done that so that the homologous sequences are aligned, you just do the crossover and see what happens. Okay, and what happens is basically deletion of the region between the repeats within the part that was involved in the crossover, and then a loss of that information from the chromosome itself. Another way of showing this type of inverted or type of twisting is here. This is essentially redundant with what I just said, that the little blue pieces are repeated sequences, the chromatid twisted around in order to allow recombination between them. And if you follow the recombination event, you will see that you have a little circle that's lost from the chromosome and a chromosome that's left with just the repeated sequence, one copy of it. Yet another example of the same thing. The figure on the left shows the arrows are the repeated sequence. A, B, C, and D are to show you what happens when you align and recombine those. So you align them, do the recombination, and you find out that you lose the region, including B and C, and you're left with one copy of the repeat, which includes A and D. All right, so recombination between LTRs can lead to the formation of solo LTRs. So I'm going to illustrate that here. Okay, so here I've shown A, B, and C representing the LTRs, and you can see they're in direct orientation because the one on the left is reading ABC, and the one on the right reads ABC. So we align our chromatids by our chromatid with itself by twisting it around, and then we make a recombination event within the LTR, between the repeated sequence. So what happens is, of course, you have a recombination event. Start with to the left, go through AB, cross over to C, and continue to go to the right. So you can see that you end up with a chromatid, which is now missing part of the information that between the two repeats and the region in between it. Okay, drawing that here, I'm following the recombination in yellow. The previous gene, AB, cross over to C, next gene, and that leaves the chromosome missing some of the information that it used to have, including one copy of the repeat. Now in pink, I'm showing the part that's deleted, and that was what's left over after the crossover event occurred. So this type of recombination leads to loss of information from the chromosome and a deletion of sequences, and is believed to be a major reason why we've got so many solo LTRs, which used to be part of endogenous retroviruses. So we've got a worksheet for practice, which is essentially the same, only we're using ALUs instead of LTRs, but the idea is exactly the same. We have a gene, which includes ALU elements within its introns, which is not exactly a rare situation, and we've got 3 exons. So we can actually put these ALUs through a recombination event, which will lead to the deletion of exon 2, and that's our question. Okay, one easy way to do this, of course, is by unequal sister chromatid exchange, where we're going to exchange between ALU, we're going to shift it over such that the ALU, which is between exon 1 and exon 2, is aligned with the other ALU, the one that's between exon 2 and exon 3. And if we have that recombination event, we're going to end up with a chromatid which has exon 1 ALU, exon 3 with exon 2 missing. Now the reciprocal event ends up being a duplication of exon 2, because if you follow that, you get exon 1 ALU, exon 2, and then the ALU recombination, and then you get another exon 2, ALU, and then exon 3. Now this is sister chromatid exchange, and that does not lead to deletion in the same way to a chromatid, but you still get the event where you're deleting exon 2 from one of your chromatids. So the other way to do that is to align the alus the way I've drawn in red. The little red things are the alus, and the arrows indicate their direction. And a crossover event between them you can follow with the green chromatid, starts in exon 1, and then crosses over. The looped out part is exon 2, and then you get to exon 3. And of course, the looped out part ends up separately as a circle and is lost. Okay, so the second type of event is when you have recombination between inverted repeats. And that's a different situation because you don't have to twist the chromatids around quite so extravagantly, but you do have to pair the repeats such that they're lined up ABC, ABC. And that recombination event will not lead to loss of information, but it will lead to an inversion of it, which can be devastating if it occurs in introns within genes. Okay, so this is an example of a real mutation that really does occur in human populations. The repeated sequence is called F8A, and you can see two of them in this factor 8 gene region. One is upstream of the promoter, and the other is within the gene itself, within an intron between exons 4 and 5. Notice that those are arranged in an inverted orientation, meaning they're pointing at each other rather than being pointing in the same direction as each other. So the first thing we need to do to get this recombination event to occur is to pair those two so that they can actually undergo recombination, and just then look at the outcome. So I'm pairing these for recombination by making this little lollipop shape, where the red arrows represent the repeated sequence, the F8A sequence. So we can follow this by, again, starting at the left, crossing over where the crossover occurs, follow the loop around, and then look to see what the outcome is. And what the outcome is an inversion of the sequence within the loop. Okay, so I'm following this around with little arrows in yellow, and we're not going to lose anything, but we're going to flip over the orientation as a result of this exchange. So the ultimate, after you've done that, is instead of having F8A promoter, exons 1, 2, 3, 4, etc., F8A, we have an inversion where we have F8A, and then we have the exons 4, 3, 2, and 1, then a promoter, then the other F8A, and then the rest of the exons. So this promoter could drive expression of exons 1, 2, 3, and 4, but exons 5, 6, going up to, I forget, maybe 20, that would be lost. So this gene is totally messed up because it's got an inversion within it that was promoted by the repeat recombination. So that's not a good thing to have happen. Okay, one more short thing that I want to just mention so that you've heard of it is a process called gene conversion, which can result from, again, alignment of not exactly identical sequences. And then as part of the recombination process, there are a lot of strand exchanges and massive DNA synthesis near the site of the break in the DNA that initiates the recombination. And sometimes this does not lead to chromatid exchange, but rather a process called gene conversion, where sequences within one allele are basically copied to the other allele, causing the other allele to be converted or changed to match the first allele. So this is not a process we need to deal with in detail, but I did want to make sure that you'd heard about it at some point. So the idea is that you've converted one allele to the other, really as a result of degradation and replication of DNA at the recombination site. So you can get changes from one allele to the other as a result of this process.